radio waves, right? And in this electromagnetic spectrum, there is a region which is called as visible region, in which we are able to see the colors, beautiful colors which are available in our nature. We are able to see the blue color of the t-shirt, of your shirt, just because it lies in the range of visible region, right? Similarly, as we all know, white light, when we pass this white light to a prism, it splits into a 7 colors, right? And each 7 colors has different wavelength, which is shown here. The red color is having a higher wavelength. Similarly, as we go from red to orange and to violet color, the violet color is having a least wavelength, right? This is a basic about light. And light is most important in terms of fluorescence, because when we pass our light to the molecule, what happens? What happens? A molecule absorbs that light. When it absorbs that light, it allows a molecule to, every molecule has an electron in that, right? It goes from ground state to high energy state, right? Excited state, exactly. And after absorbing that light from ground state to excited state, it emits that energy, because everyone wants to be there in the ground state or the stable state, right? After coming from excited state to ground state, it emits a light in the form of electromagnetic radiation that is called as fluorescence. And how it was discovered? It was discovered by Dr. W. Harshal in 1845. What he found, that when he took a tonic water is basically a quinine solution, a chemical molecule dissolved in a solvent. It makes a quinine solution. When we take this quinine solution, it is always a transparent in color in visual light, right? When we pass this UV light on the quinine solution, what he found, that the color of the quinine solution was blue in color. From there, he found that this phenomena exists in our nature. Next comes another scientist known as Sir George G. Stokes in 1852. He coined this term fluorescence when he found that the gemstones also shows fluorescence when it is exposed to a UV light. From this, the term fluorescence was coined by the G. Stokes. And everyone says that it's a magic, but it's not a magic. It's a basic science. Right? Behind that. And how does it work? Basically, what I told you, the electron beam or laser beam is exposed on the molecule. It allows an electron from ground state to an excited state. And then there is the emission of the light from excited state to the ground state. This pictorial representation is known as Jablonski diagram, which was discovered by Alexander Jablonski. This is a detailed Jablonski diagram, where various processes exist in the Jablonski diagram. When we allow light, the electron from the ground state, it goes to various excited states. There are various excited states in a molecule. There is the dissipation of energy, because electron is not a static electron. It always revolves, or it has a motion in that. It takes various steps. What happened? It takes internal crossing, and then there is a triplet state in the molecule, where it goes to in the form of intersystem crossing. But when this electron, these are all excited state. And this is what? A ground state, a stable electronic ground state. It comes, when this electron comes to this ground state, this is known as phosphorescence or a fluorescence. Phosphorescence, what happens in phosphorescence? It comes from triplet excited state to ground state. But in fluorescence, it comes from singlet excited state to a ground state. This is the difference between the fluorescence and the phosphorescence. And various relaxation processes are there in the fluorescence. And in nature, as we all know, we see various bioluminescence or fluorescence process in nature. A simple example is fireflies. Everyone knows Juno. Yes. That is an example of bioluminescence, where a molecule is getting oxidized and it forms a fluorescence color. It's observed in the Juno, something like that. And seashore में भी देखा होगा, बहुत सारे reels आजकल तो Instagram में सब send करते something ऐसा. Now, that is the fluorescence observed in nature, right? And how do we see fluorescence in synthetically or man-made fluorescence? That is a chemical molecule, right? That molecule is responsible for emitting light when it is absorbed or exposed to a UV light. Similarly, these are the molecules which shows fluorescence in nature. Simple example, what we can see is having this. You will also see here various examples of a chemical compound which is having normal color in UV light and different color in normal visible
light.
Right? So why do we see this fluorescence colors? Just because of the various pi bonds. Have you all know about pi bonds?
Yes.
Right?
Yes.
Because of the various pi bonds present in the chemical molecule, and there is electronic transition happening in the electronic molecule, molecule compound. Due to this, we see a fluorescence. Next, these are the fluorescence molecule core structure, which shows various derivatives of these compounds shows different fluorescent color. As we all know, naphthalamide, acridine, phenanthridine, indole, these are having inherent properties of being fluorescent. Now, and what is the use of this fluorescence in our life, right? Because in nature we see various examples, but why do we synthesize these organic dyes? Because in scientific world we use this as a fluorescence sensors. What do we mean by sensors? The sensors are those which can detect something, right? If we wave our hand like this on an electronic instrument, it is detecting our wave or direction of the movement of the radio waves, whatever it can sense or something like that. It is sensing, without touching or something like that. Similarly, in chemical molecules also, they can also act as a sensor. These are having this is the chemical molecule, right? When it is exposed to a metal ions, just say cadmium metal ion, which is a toxic metal ion water present, right? It can detect and it can show different color. This is a visual light color which you can see. Starting the probe color was blue in color.
Blue.
As soon as it get exposed to cadmium, it showed green in color. See, various metal ions we have tested over here, and particularly it was sensing to a cadmium, right? This is the example how a molecule can be used as a sensor, as a fluorescence sensor. Also, there is another use in biological studies for staining various cells just to know where is the problem in our cells, cancerous cell hai ya kya hai, something like that. So in nucleic acid detection, what happens in that, we are having a probe and it interacts with the DNA or RNA of the molecule of the cell, and it glows in as a fluorescence, which can be tested by using gel stain. Gel stain, we perform an experiment called gel stain. There we see a fluorescent color is observed or not, whether it is binding to that DNA or something or not. Next, this is our RT-PCR kit. You all might not be knowing. You are not knowing. COVID kit, which was in-house developed in our lab in CDRI, and it is also marketing available to detect COVID and other diseases. And in this RT-PCR kit, various quenchers as well as fluorescent dyes are available, which can be used for the staining of detection of the COVID or something like that. And thank you. These are